We've been talking about DNA replication and using this depiction of the replication fork where you have one leading strand and one lagging strand, which make up the replication fork. But remember, at the other end of the replication bubble, you also have a leading strand and a lagging strand. So one strand of DNA can be the leading strand and the lagging strand depending on its orientation with respect to the origin of replication. Replication of the leading strand is less complex, and you might expect this to be faster. So a key question is, how can replication of the leading strand be coordinated with replication on the lagging strand at each replication fork? Recall that there is a DNA clamp associated with DNA polymerase that keeps the two strands together behind the DNA polymerase as it moves along the strand that it's copying. This clamp must be loaded onto DNA, and there is a loader protein complex for the DNA clamp. That loader complex serves to place clamps on both the leading strand and the lagging strand, and it holds them together. This results in coordination of the movement of the replication fork between the leading strand and the lagging strand. This clamp complex, which requires energy in most organisms for the action it does, prevents the leading strand replication from outstripping the pace of the lagging strand replication. So you get coordination of leading and lagging strands. But this coordination raises an interesting question. Why is replication only 5' to 3'? Why can't there just be 5' to 3' synthesis on one strand and 3' to 5' synthesis on the other strand, so that it could be easily coordinated? The reason is that we need to be able to proofread that added nucleotide, and this is only possible in the 5' to 3' direction, using the materials that we have. The chemistry of the reaction requires that the orientation of synthesis be from 5' to 3'. The release of pyrophosphate from the incoming nucleotide and the subsequent hydrolysis of pyrophosphate to phosphate provides the energy that drives synthesis. Without this, the process stops. Overall, you want the reaction to process forward, but with high fidelity. You want it to have the proofreading capacity, but you also want it to be processive. To understand this, let's look at a comparison between 5' to 3' growth on the right and hypothetical 3' to 5' growth on the left. In both of these, pyrophosphate is released as the incoming nucleotide is added. However, on the left, pyrophosphate is released from the end of the growing chain to create the phosphodiester linkage, whereas on the right, it is released from the incoming nucleotide. So in both cases, we can drive the reaction forward. But now, let's look at the proofreading process. If an error is made, the nucleotide is removed by the 3' to 5' exonuclease activity of polymerase. The difference is that on the left, nucleoside triphosphate is removed, and on the right, nucleoside monophosphate is removed. This leaves a 5' monophosphate on the left and a 3' hydroxyl on the right. Following removal on the left, there is no longer a triphosphate at the 5' end of the chain to release pyrophosphate and drive the formation of the phosphodiester linkage. Polymerase stalls. On the right, this is not a problem. The new incoming nucleotide brings a triphosphate group that can be hydrolyzed to drive the reaction forward. The ability of polymerase to proofread and continue adding nucleoside triphosphates doesn't work when the high-energy bonds that drive synthesis are removed during proofreading and not replaced. Synthesis in the 5' to 3' direction overcomes this because each incoming nucleoside triphosphate brings with it the stored energy to add to the growing DNA strand. So the proofreading process only works in the 5' to 3' direction. But is there an alternative? Can you think of a way that you could actually grow 3' to 5' instead of 5' to 3'? What would make it work? Could you imagine a cell where 5' to 3' and 3' to 5' synthesis were mixed? If we look at the replication machinery itself, all of these enzymes that are involved in this very complicated process are brought together to form a complex that is functionally a machine. It is a machine that copies DNA. So you have all the necessary enzymes, factors, everything that you need at the same place to carry out this process. And that was sort of implicit in some of the interactions that we discussed last time, such as between primase and helicase, between DNA clamp and polymerase. Bringing all of these together because they need to act at the same place at the same time would be very important. The overall mechanism is similar in eukaryotes to what we discussed, which is based on prokaryotic mechanisms, but there are more components. There are more subunits to the DNA polymerase, for example. But the fundamental processes that we talked about are similar. Why would having the proteins involved in replication, especially initiation, associate with one another be useful for the cell? Think about the enzymes and proteins involved in DNA replication. What types of interactions would stabilize the formation of these higher order complexes?